with going over the stuff we've been working on with the Institute, this was just from the introduction slide, sort of implementing the idea that today we're going to be talking a little bit or mostly on the bottom end of this, thinking more about what's going on with molecules and sarcomeres. But I think we all know that's related to much larger scale in both directions. And there's a lot of interest in that from everybody that's involved here. And so as an overview of what goes on in my lab, we do a lot of single fiber mechanics, mostly with skin fibers. We do a tiny bit of intact muscle work, but not very much. And we also do some computational modeling. So we try to integrate the computational modeling and the biophysical experiments to investigate the molecular mechanisms of contraction. And so just as a very small introduction, a lot of the proteins that we're going to be talking about modeling today are shown here at the bottom of the slide, where we've got the thin filaments that are made primarily of actin shown in blue there. And then, do you guys see my cursor that's moving?
Yeah, sometimes.
Sometimes. Okay.
I don't see it right now.
All right.
Oh, yeah, there we go. Yeah, I can see it now.
So it's
It's
down on the
white,
thick.
so when the background is white, it's hard to see, but I think it's fine.
Okay. Thank you.
Ah,
There's the thick filaments of myosin shown in red, and it's the actin and myosin interactions that generate the force and shortening to power muscle contraction. And so also associated with that are some regulatory proteins, and those can be on the thin filament, like the troponin and tropomyosin, which are shown in green and pink. And then on the thick filament, there's some regulatory light chains on the neck of the myosin crossbridge. And there's also the myosin binding protein C and the C zone of the sarcomere. And so those can affect how actin and myosin interact. And we've done quite a bit of work trying to understand those crossbridge interactions and how to model them and how to measure them for some of the kinetics. And as an overview, I've got some introductions into some of the history of muscle modeling over the years. And then I've got a couple examples looking at some spatially explicit modeling. And at the end, I've got some code that I can run for you, or you guys can run together if it works. It's in the folder that Victoria had sent out. It's like very stripped down and basic, but I think that might make it easier to talk about some of the simple parts that we're trying to interact with. And so to get started there, computational modeling in general can go over a bunch of different scales, like we showed just a moment ago on that slide. But usually in most general sense, computational modeling is trying to better understand computing and mathematics and theory to try to study complex systems and how biology works across different organizational scales. And on the most simple level, I just thought maybe we could start with a biomolecular reaction. Imagine we were mixing two different things with substance A and B, and that's got some rate K that goes into C, for example, to make a third substance. And so you can write some differential equations that describe this. And so because A and B go into C, you've got that description of how that changes in time. So the rate that occurs times the concentration of A times the concentration of B will be how C changes in time. So that's how fast C gets created. And then similarly, the negative of that is going to be consistent with how fast A and B get depleted. And that's what that D of A over D of T and D of B over D of T are the negative of what is the concentration increase over time for C. And so if you solve those equations for the different A, B, and C substances as a function of time, you see that depending on how much A or B you have, it decreases with time, and that turns into C, okay? And as an example, you could imagine that this, how much C you have depends on how much A and B you have, and that's what's kind of shown over here on the right, is as you get increasing amounts of A, you would get increasing amounts ultimately of C. So that's just a very, very simple example of trying to use some math, and technically that's a model to describe how your system is responding. And so quite a few years ago, some of the earliest models to try to describe what was going on with force production was from A.V. Hill, and he shared the 1922 Nobel Prize for linking heat production and mechanical work in muscles. But he came up with a model that has been widely used for many, many years, and I'll talk on the next slide about some of its properties. But essentially, there was this passive element shown here on the left, and then there was this contractile element representing what muscle does to contract, and that was in line with a series element, which you could think of, there's a passive and a contractile element of muscle, and that series element might be something related to tendons or might be something related to other components of the system. But essentially, that led to some force production that was a combination of all of those. And so here shows some of the characteristics for imagining you were simulating or measuring a muscle twitch, or the amount of force that is generated with a muscle. And so that's on the y-axis, and on the x-axis, that's relative to the length of the resting muscle. And so they've shown here that resting muscle length is 0. And so this happens to be a component where your passive component is very, very low until you start to stretch that resting muscle, and that's shown in blue. And then the active component is sort of overlapped with the green and the red at these shorter lengths, but this starts to show you that active component increases as you're increasing length, and then there's some flat phase there, and then that starts to decrease. And this sort of looks like that ascending limb, and then the flat component and the descending limb of the force length relationship. And if you add the blue and the red together, that gives you green. And so that's why green comes up and then down and then starts to go back up, because at very, very long muscle lengths, you start to change that overlap. And so you're decreasing how much force is produced by the cross-bridge interactions, but you're also stretching the muscle, and those passive elements start to, like titin or collagen, or depending if you have tendon in the system, a lot of that starts to take over and increase how much force is being generated or measured from the passive elements. So there's sort of a trade-off there at the longer lengths. And this concept has been very, very useful for many applications, even though it sort of is just a simple description of how forces change with the length of the muscle. And so the next real big iteration in this was trying to understand, maybe on a smaller scale, what might be going on at the molecular level. And so this came out of Andrew Huxley in this really classic 1957 paper, and there was this is a very long paper, but there's a section of it that he puts forward some ideas about the cross-bridge theory that ultimately becomes like cross-bridge theory and actin myosin binding interactions. And so there was this idea, perhaps, that you've got your myosin filament and you've got your actin filament, and they're sliding past each other as the muscle's shortening. And maybe there's some sort of an interaction here, which is shown by this little M in between the springs, where this M, being myosin, is trying to bind to A, actin, and depending on how fast they're going past each other and how close M and A are together, that might change how much force is produced. And so it was this idea of some sort of spatial distribution of interaction between the molecules. And so they described how those interactions might be with this stuff, these rates of binding that are called F, and that would be like how that A and M interact to go from an unbound state to a bound state. And then that detachment event would be what they call the G for the rate constant G, and that's for detachment. And they made these dependent on the position or the distortion or how close that A and M were, where if A and M were very, very well aligned, and there wasn't any stretch in these springs, it would be a very low force dependent, or it would have an interaction, and then that increases, the rate increases as you get closer and closer and closer to this H. And that H is sort of defined on how close X and H are to stretching the springs. And I'm not saying that very well, but the idea is that there's some position dependent binding, and then the detachment events, depending on if you're stretching them, have a different rate that is position dependent. But if they're going past each other very quickly, so your H is going negative, then you see this G is very, very high. So if you're stretching one way, you're producing force with less of a rate to detach. And if you're strained the other way, you're going to detach very, very quickly. And so what came out of some of those ideas, which this little thing in the top is in the way, I apologize. But what you're seeing is when you're not moving at all, or there's no sliding between the filaments, there's a high level of binding, or a high amount of force that's generated. And then as you start to shorten faster and faster and faster from each other, so as your velocity of shortening is going, you know, from 10 Vmax to 25% Vmax to all the way Vmax, two things start to happen. Number one, you sort of get these different distributions of binding in terms of how much force can be produced depending on how far those springs are stretched, but also how much force is produced as they're sliding past each other. So where up here, there's no force produced in the negative stretching direction, here there's some little bit of force that's produced, and this starts to flatten and smear out over the distribution. So there's a lot more information in the paper about what the implications of this would be, but this sort of is some of the foundational theory about how these molecular interactions might occur in muscles. And so this was a nice summary figure from a review written by Walter Herzog, where it sort of shows the evolution of what people have thought about over the years. And so this is what I just described to you. But then in the 1970s, the Huxley and Simmons had a 1971 paper where there was sort of this idea of a cross-bridge sort of ratchet or position in head, or movement of the head. And then that's sort of summarized nicely here with some of the atomic structure that came out of a X-ray diffraction model from Ivan Raymond in 1993. So where they're sort of showing that as ATP comes in and hydrolyzes, then you come around the circle and you bind, and your cross bridges bind. And then there's this sort of a power stroke thing. If you look here at the bottom in number 4, the neck of the cross bridge is sort of like this, and then it goes like that from 4 to 5, and this would be the force generating event. And then as you detach, you get this sort of repositioning back to where it was originally with the hydrolysis. And so some of these ideas sort of all came together over the years for what people try to use and put mathematics to describe how muscle might work. And so the spatially explicit modeling is even one level of sort of system interactions higher than just the actin and myosin crossbridge, where we're trying to simulate what's going on inside of a crossbridge, inside of a sarcomere. And so most generally, you could imagine you've got this thick and thin filament interactions in a system. A lot of what I'm showing you today is all modeled in a half sarcomere, and that takes advantage of some of the symmetry around the M or the Z line in terms of what a sarcomere would do to generate force and shortening, where you only can model half of it and assume that's what the whole sarcomere might be doing. So then some of the inputs to that model would be perhaps calcium or perhaps sarcomere length, where you've got changes in the overlap. And some of the outputs you might be interested in modeling might be the force that's produced, or how fast actin and myosin are using ATP. So if you're interested in energetics, or maybe you're interested in how many cross bridges are bound, or how much of the thin filament is available for binding. But within all of that, there's components to try to describe what's going on in the sarcomere in terms of what you're trying to describe with the geometry or the kinetics or the mechanics. And a lot of what led to this spatially explicit modeling had a lot to do with the mechanics. And this is sort of a subtle assumption that I'll try to get across today, but there were a lot of measurements that were in the mid-90s from some X-ray diffraction measurements that started suggesting that the filaments were actually stretchy and extensible, because over many, many years everybody had assumed that the filaments couldn't stretch and that everything went on was just the actin and myosin interactions. And so if the filaments couldn't stretch and it was just modeling the interactions between actin and myosin, then what you're describing with like one actin and myosin could represent the entire population. And so if the system starts to be able to stretch, then that distribution or that distortion or the springs between the position of the actin and myosin versus the neighboring actin myosin might start to become different if the filaments become extensible or stretchy. And that came out of first it came out of some biophysical measurements, but then this led to the idea of the spatially explicit modeling, trying to say, well, what happens if we stack up a system of cross bridges and binding sites in a bunch of, like, just initially this was a 2-filament model where there was a system of springs for describing how stiff the thick filament is versus how stiff the thin filament is, and then how stiff the cross bridge might be. And that started to try to test the implications of, if the filaments could stretch, what might that be an outcome from there. And so that started this idea of the mechanical properties might matter. And so the geometry that was first used by this Daniel et al. 1998 model was just 2 filaments and a simple system with 20 thick filament nodes, or where 20 cross bridges would come off of the thick filament. And these nodes are sort of shown in white here. So this yi minus 1 and yi and y, for example, are the thick filament nodes, and that's where the cross bridges extend from. And between each node, there's a spring. I know it's a little difficult to see with this particular figure, but there's a spring that is between the nodes. And then you describe a similar system for where those cross bridges combined along the thin filament, as shown here on the bottom. And so because there's differences in how far apart the cross bridges are spaced when they line up with each other versus where the binding sites would be, that's why there's 20 thick filament nodes and 30 actin binding sites, because the periodicity of the helical coil or twist on the cross bridges is 43 nanometers for the thick filament and about 37 nanometers for the thin filament. And so I'm going to go over kinetics later, but you had to describe how these interactions would occur. And the kinetics involve some position-dependent cross-bridge cycle. And it ended up that it's very, very difficult to solve a large system of equations with the ordinary differential equations or even partial differential equations to describe how this system might interact. So they implemented what's called Monte Carlo algorithms, or stochastic kinetics are driven with probabilities of binding and then that made some of the solutions to the forces a little easier. And so some of what came out of that is what I've got going forward with how force might change depending on the mechanics of the system and the geometry of the system. And we'll get into a few more examples on these next 2 bullet points where I've actually got some results to try to show you. But before I get into all of that, I wanted to revisit some of the basic muscle properties and model formulations that are trying to be described by some of these spatially explicit models. Does anybody have Any questions so far on just some of the backgrounds of the modeling? Are we good? I'll go on. Okay, so this is supposed to represent kind of the end view looking down the sarcomere of what would look like the thick and the thin filaments. And part of a lot of what I'm going to talk to you about is trying to understand what might be going on with just one interaction of like one pair of thick and thin filaments versus multiple thick and thin filaments working together. And so that's a lot of the difference between some of the work I've done versus some of the original spatially explicit two filament model that I just showed you. And so, as we've all mentioned, the basic components of muscle are turning mechanical energy or chemical energy into mechanical energy. And the tenets that we try to describe there are the mechanics, the geometry, and the kinetics that might underlie this. And so there's characteristics all the way from the whole muscle down to just the myosin and the actin with how these spatial properties occur and what leads to the structure of the sarcomere that we try to simulate. And some of this, as I mentioned before, is calcium dependent, depending on the troponin and tropomyosin characteristics. And then adding on there is some of this cross-sectional geometry that we can try to say, what are the implications between just two filaments versus multiple filament interactions? And so just to describe some of the kinetic interactions that you'll see today or what's behind them has to do with some sort of a kinetic scheme where you have no calcium binding, then calcium comes into the system, then that'll activate the thin filament, and then this particular state will have thin filament being activated, but the cross bridge is not attached with the ADP and ATP, the hydrolysis after ATP hydrolysis. And then as the cross bridges bind, then they will go through some sort of a power stroke where that like neck transition changes as phosphate's released. And then that ADP will dissociate into the Riger state, and then ATP comes on for hydrolysis, and all that starts all over again. But we're actually trying to model, as mentioned just a minute ago, some of the implications of what's going on as these filaments are rearranging, and one cross bridge is pulling on another cross bridge, or pulling on a filament, and how that affects the system response. And so, as I mentioned, we're scaling up from this Huxley model for one myosin to this two filament model, and then trying to look at maybe what are some of the implications of these multiple filaments. And we always try to do the best we can to use some of the literature values or our own experiments to try to describe and predict what's going on with the model predictions versus the actual measurements to validate the model. And so a couple of examples here we'll go over in the next few minutes, and then we'll maybe get to running some code as well. Okay, so two important things to think about is we've got the mechanics, so cross bridges are elastic, filaments are elastic. We've got geometry having a component that affects kinetics where there's position dependent and strain that changes the system. And then we have like bound or unbound forces. And then this shows you scaling up from what was assumed like that well-mixed population of cross bridges and one particular interaction, as we've shown before, versus a system where there can be some flexibility and the linearly elastic springs between the different myosins, and what are the implications of that. And so this is a summary of trying to show you what people found. So this is the conclusion before I'm showing you the results, but it seems to be that this extensibility or compliance or stretchiness of the filaments seems to coordinate the cross-bridge binding. And that showed that compliant realignment from that cross-bridge binding and moving one filament relative to the other leads to increased cross-bridge binding in the system. And so the way that that goes, as an example, might be that this cross-bridge binds, and then as it goes through its power stroke, it moves the filament, the thin filament, relative to the thick filament, and that leads to more favorable realignment and more favorable binding of the next nearest cross-bridge head, perhaps. So you see that that favors binding with better realignment between the pink head and the yellow binding site. And so that was for two filaments, what I just showed you. And then it looks like then that compared to just one actin-myosin crossbridge, some of the findings from this suggest that the lattice compliant increases crossbridge binding. And then I'll go on to also suggest that as you get more filaments interacting, that amplifies or increases the relative effect of that compliant realignment that I just tried to show you. And so this is just summarizing how the mechanics and the geometry might play a role in favoring binding of the nearest neighbors. And so with the two filament model, one of the things that first came out of this was if you were changing the stiffness, so this is shown on the Ka axis on the bottom here, and the vertical axis, this T naught, has to do with how much force is produced. And then if you're changing the crossbridge stiffness on this other axis over here, Kxb, as you vary the relative stiffness values of the filaments versus the crossbridges, that leads to some sort of tuning or some sort of surface where you get sometimes very low forces that are produced, because that could be there's either very little crossbridge binding, or on a very compliant system, for example, when you have very low crossbridge forces, you can get very low force production even though you get high crossbridge binding. And then there's some sort of sweet spot where you can get high force production, and that leads to where I put this little white star is sort of on the shoulder there, where you're getting pretty high force production and pretty decent crossbridge binding. That would be equivalent to the rough values that are thought to exist in the muscle. And so the next thing that we went on to ask a little bit are, well, what is the implications here of that ratio of filaments? It's not just a one thick and one thin. There's actually a ratio where there's two thin filaments and one thick filament, where essentially every thick filament interacts with 6 thin filaments, and every thin filament interacts with 3 thick filaments. And this is also a calcium regulated process. And so the first step that was tried to look at this was a paper by Bryant Chase, where they tried to look at a system that was sort of as shown here, but there were some complications with what would go on with the edge effects, because it didn't have the boundary conditions were different on the outsides versus where all the thick filaments were interacting with the thin filaments on the inside of these hexagons. And so what I tried to do was figure out a way to get rid of those boundary conditions for my PhD, and we put together some ideas of how to represent and how to model this, trying to use that physiological ratio of thin filaments, adding in the calcium regulation, and then that would lead to something that was a 3-state cycle, as I showed you before, for thin filament activation as well as crossbridge binding. And one of the things that helped was putting where this dashed line is shown here on the right, is we simulated 4 thick filaments and 8 thin filaments, but we only simulated the interactions that would be within this dashed line. But at that dashed line, we made periodic boundary conditions such that what would be going on, for example, on the other half of this thick filament would be occurring right here. So we were sort of wrapping the edges onto each other, and that removed some of those edge effects of that prior model from Bryant Chase that helped simulate what would be infinite lattice space with just a finite number of filaments. And so then this shows you how we've got same structure as I showed before, where you've got a thin filament made of the thin filament nodes with springs between each node, and you've got a thick filament made of thick filament nodes with spring between each node. But then in a collinear plane, as shown in the slide there, we would have then two cross bridges coming out of each node to interact with those same collinear planes of the thin filaments that it's facing. And that would occur as you'd move throughout the system radially as well to try to simulate all those interactions that I just showed within this lattice space. And so to show you some of what we found with that, I was going to skip over this because we sort of showed you before, unless there were questions, that's sort of there as a placeholder. And we saw that physiological filament geometry amplifies those interactions of the cross bridges and the compliant realignment like I showed before. And so if we use the same exact model and only allow for interactions between this row of actin and myosin as outlined by the oval versus the whole rest of the system, you saw that there was a whole lot more first produced with the multiple filament model versus the two filament model. Except that might be what you'd expect because there's so many more interactions. So if you then even go and scale that to the relative force in terms of the amount of force produced divided by the myosin, this is what the relative forces look like as a function of time on top for A. Then number 2, or panel B, is the thin filament activation levels. And then the bottom is the cross-bridge binding as a function of time. And then this is what's shown here is for 3 different PCA levels. So if we thought about, we see that the fractional activation is very similar between the two models. And so at PCA 4, you have a lot more activation, which is a high calcium level. At PCA 6, you have medium level of activation, which is a medium calcium level. And at low calcium, you have very low activation. And so even though the fractional activation is the same, you see that you get very, very different relative values of the force that's produced per myosin in the system for the multiple filament model, where that's quite a bit larger. And that comes from quite a bit more cross-bridge binding as well. And so one of the take-home messages is that you get more binding and more compliant realignment with the multiple filament model. And some of the implications of that would be then at PCA4, that predicted relative force would be about 2 piconewtons per myosin versus about half of a piconewton, or sorry, 2 piconewtons per cross-bridge instead of about half for the two filament model. And that seemed to line up, it turns out, a little bit better with what was been measured with single molecule measurements. And then if we thought about what was the differences between the fractional myosin binding, you saw that there was greater cross-bridge binding and there was greater cross-bridge turnover going through the cycle faster. And so all of what was going on with the forces, the level of binding, and how the dynamic turnover of the system was happening seemed to be much more in line with some of the measured values from muscle experiments for the multiple filament model, trying to simulate or emulate the regular physiological geometry compared to the two filament model. And so that seemed to be more consistent with the experimental literature. And that was one finding that came out of the geometry amplifying or coordinating cross-bridge recruitment and cross-bridge cycling. And so it seems like that coupled cross-bridge activity depends on how many myosin are able to generate force, which depends on the geometry or the system structure. And so that's sort of the end of the results of what I wanted to show you. I've got a little bit of a wrap up of some conclusions and some other models that have tried to implement some other things with this idea of the spatially explicit modeling. And then I've got this two filament code example, if we'd like to go over that, or if anybody wanted to try to run any simulations, or I can run them on my screen and we can talk about them there too. So in conclusion from what I showed you a bit today, these mechanics, geometry, and kinetics are all linked, and they're hard to separate, but they're also a little bit difficult to model with these larger systems compared to some of the simpler models I showed you before. The physiological geometry seems to coordinate cross-bridge activity and increase the, this should be thick filament binding, not thin filament activation, because we saw that the thin filament activation was very, very similar between the two systems. So this is a typo, I apologize. And then those cross-bridge dynamics seem to scale with the population of what's going on with the cross bridges that are generating force. And so all of what I showed you today assumed that there were only interactions within one direction. And even though I was kind of showing that they had interactions sort of vertically between the filaments, I really only measure all the forces and calculate the forces in one dimension. So some people have tried to move forward with saying, well, what might the implications be of, rather than just having all the forces balancing in one direction, cross bridges actually sort of have this weird sort of movement of both a swing and a transition as they go through that has a vertical component or a torsional component as well as a longitudinal component. And so there's been some models that have tried to estimate or model what goes on as the distances between those filaments might change, which usually occurs as you change the length of the muscle. And then some other models have tried to implement some of the components of the passive elements, such as Titan, for example, where Titan runs along the thick and the thin filament here, and trying to say, well, rather than just having the myosin just be there at a free end, there's actually Titan that ties it to the Z disc, and they put a spring constant in there for Titan. And you could see that depending on what's going on with the passive forces, and you're changing the length of the sarcomere, the effects of Titan have a significant impact on what's going on with the passive contributions, like we had shown even with those Hill models. And so other folks have said, well, what might be the implications of myosin binding protein C, for example, because it's thought that myosin binding protein C stabilizes or doesn't let the cross bridges bind as well. And so they put together some kinetic models where myosin binding protein C, which is in the C zone of the filament, it competes with trying to hold down myosin versus allowing myosin to bind. And so that's one example that came out of this FiberSim model. And then another group has tried to simulate twitches, and Simon presented a workshop earlier, and they tried to look at what was going on with trying to simulate twitches and trying to simulate work loops to try to understand some of the differences between the stresses and the strains and the energy use and simulated work loops. So those are just some different applications of these ideas of the spatially explicit model. And part of this, and part of what we're ultimately trying to work on as a group, is bridging over these different scales to try to better understand what might be going on down here at the molecular and sarcomere level, with trying to scale that up to better understand what's going up at the, with the muscle tendon architecture, or the effects of the excitation contraction coupling or neuromechanical control of locomotion. And so some of these ideas are trying to see how you can go across these different scales, both mathematically and with some of our experimental measurements. And so that's essentially the end of the presentation. And if we wanted to go into some of the two filament code examples, I don't know how it's best to start to do that. I've got some additional slides that describe in more detail some of what the code does and some of what the mathematics are using to describe even just a simple two filament model. And I can also, so I can go through those, or I can also just run simulations Does anybody have any ideas or suggestions on next steps?
I think it'd be great if you walk us through the simulations. If you,
That sounds good. So, and I can walk us through a simulation, and then I can also show you some of the, like, so I've just got to show you the slides that I have that we can come back to. I tried to describe what goes on with like balancing forces in the system, in terms of when cross bridges bind, what happens, and then some of the math behind that to balance those forces, and why we use linear algebra. And then I just wanted to have another snapshot of the kinetics if there were any questions there, although what I have today for the two filament example doesn't have any of this stuff on the left side with any calcium regulation. It's just sort of the cross bridge interactions. And then I also wanted to make sure to put in there this whole sort of concept of these in the spatially explicit models, because there's positions on where myosin and actin can interact and bind. When the cross bridge binds, sometimes it can be stretched out quite a lot, and that might be a low probability of being stretched out to bind, or it can be like in a resting position where there's a high probability of binding. But the point behind all the kinetics is they all depend on distortion of the cross bridge head. So rather than just like a rate constant, it's a rate constant as a function of distortion. So they're actually rate functions. And so like this would be an example. This is sort of looks like a Gaussian function for the attachment. And then these are sort of position dependent power stroke or detachment events that I just wanted to make sure were resources that were there if we wanted to come back to them. So I need to end this for now. And so if I were to in like the research workshop folder that Victoria had sent out, if you were interested in downloading any of this stuff or seeing how it worked for yourself, if you click on that computational modeling link, these are all MATLAB files. And to summarize, like I wrote a very basic sort of summary of what was in there. And essentially what I've put forward is what is a set of code that's very similar to that code that was used for that two filament model published by Daniel et al. In 1998, where there's links to the paper that are right here. The main code that I'm going to run for you is this called 2fil MC blah, 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 right? And then that calls some scripts and functions within it where there's a parameter file, and I can vary some of those parameters and we can see some of the parameters, what the different outcomes are. There's this build M core model, and that build M core model has a lot to do with trying to line up all the positions of like all the nodes and who interacts with which other node and how far apart like node MJ would be from AI if you're thinking about this node and this particular cross bridge that's shown by C. So that is a pretty complicated thing to go through, but that's what keeps track of all the interactions. And then there's a kinetics rate function, which that describes what I've shown here in terms of like who's interacting with whom. That gets called quite a bit. And then at the end of the simulation, I just fit the response to an exponential function. And you can totally comment this out if you don't have whatever toolbox is required to do the optimization to minimize that function, which I think is the optimization toolbox. And so that's a summary of what's in that folder. And so if I go and look at MATLAB, for example, this is the two filament MC. And if I were to just run this from scratch, I get something that pops up where I made two figures. And I don't know how well you guys can see these. Do you want me to make them bigger? So
Yeah, bigger would be a little bit. Yeah, that's
this figure happens to do with the mean force that's produced. And the reason I say it mean force is I'll show you in a moment that I run the model over and over again and then average the forces across each run. And this little exponential fit here is shown in the black line, where the asymptotic value would be how high things get on the flat asymptotic side. That should have units equivalent to piconewtons in this example. And I also keep track of whatever that rate would be in terms of the rate to the half maximum, which should be in units of per second. So this is a little bit sloppy there for all you code aficionados. And then this is just the mean of the fractions of the heads that are bound. And you can see that whether you're looking at force or whether you're looking at the heads that are bound, they're pretty similar. But I just wanted to show a couple of the outputs, for example. And so one thing to imagine, if I were to, I swear I had it in here, but maybe if I were to look at what this parameter file were, for example, right? Say we wanted to only run this one time. If it's not going to average anything, and just show you like what one run might look like, okay? Oh, I might have to save that. Whoops. So here you see that this is like much more variable, right? And so as you see your forces binding, this is sort of very choppy by comparison to the averaging that I showed you before. And part of what's interesting is if you think of like how the forces are balancing every time step versus what's going on with just the cross bridges, right? So this essentially is telling you almost, so there's a cross bridge binds here, and then it stays bound, but then another one binds, but just for a second, and that goes off, and then you have one bound, and then another one comes on, but now it seems to stay on, and that recruits a neighbor, but then the neighbor comes off, and that recruits a neighbor, but then the neighbor comes off, and now a cross bridge is, that third cross bridge seems to stay bound, and then maybe the fourth one comes on, as an example, right? So that's what's going on, is this is showing you how the cross bridges are coming on and off. But as you go in time, you're starting to stretch out that filament as you're producing greater forces, and that stretching out of the filament is starting to recruit additional cross bridges. But the reason that I had run it, for example, like, 25 times is it seems to average things and be a little more easy to interpret. But you can change that, or I can change that however you want me to. And like, so if we go back to that original simulation as an example, right, you see that our level of force we're producing is around 18 piconewtons. And if we were to maybe say we wanted to see what would go on with making the spring constant of the thin filament, like a tenth as stiff, what might happen there? here you see, even though the numbers not hugely different, you get a little bit less force because a lot of the energy is going into stretching out the filaments rather than actually generating forces. And I had forgotten, you know, in terms of the bound cross bridges, that looks like it's about 14% or something. Which I had not remembered what it was before, but I think that's greater cross bridge binding, even though you're getting less forces. As like one particular example. So those were just a couple things if, you know, anybody wanted to play around or see how that would work. That code is available there, and the code that's available there is largely associated, where's that PDF, with this particular publication that was just the two filaments. So I don't know if anybody has questions or if you want me to go through some more stuff or if we wanted to talk about anything. But that's mostly what I had prepared for the presentation side of things. So there's lots of different levels or versions of computational modeling, and that's one example. And I tried to show a few examples as the sort of historical progression of some of the ideas in computational modeling of muscle that are used.
So Bert, I guess I have some questions. So maybe to start with, you know, there's this whole idea of thick filament activation now. That's fairly new, and I think so to what extent, like, could you modify your model to include, you know, new ideas about like super relaxed, and I mean, is that a direction that you think people might take this, or?
Well, so I'd have to think about which model that was used, but there was a paper published in 2018 from Ken Campbell, where if you give me a second, I could probably almost pull it up. But there's quite a few ideas about how to model that. And it largely comes from trying to think about a transition, potentially, whether you're doing a spatial model or not a spatial model. The idea would be that, you know, in the super relaxed state, the heads are maybe down or laying on the filament or not able to interact with the thin filament as well, whether that's totally stabilized or something in the middle. It's clearly different than like the disordered relaxed state. But there's the idea that that transition between the super relaxed state to a disordered relaxed state, where then the myosin could interact with actin, has something to do with force. And whether that's force in total in the system, or whether that's the local force right at like a cross-bridge node itself in a spatial model, the concept would be that whatever that rate transition would be coming from the disordered relaxed state to the super relaxed state, or sorry, I messed those up, super relaxed state to the disordered relaxed state, that transition would have something to do with integrating the force that is being measured in the system. Or if you were doing some sort of a population cross-bridge model, it'd have to do with probably how many cross bridges were bound. If you were just doing a cross bridge model, like as the level of cross bridges increase, you'd imagine that that would recruit or bring on more cross bridges from the super relaxed to the disordered relaxed state. I think in the spatial model, you'd probably want to try to, I would think, to try to have that be dependent on the sort of strain or the stress along the intermediate portions between the nodes of the thick filament, because that's where you'd sort of have the thick filament feeling those forces, or what would be where the thick filament would be stretching out that could destabilize that super relaxed state to some degree. So it would be putting into the kinetics something that was a mechanical term in addition to the spatial or distortion dependent term, like I showed with some of those rate functions. So it would be adding a parameter that would have something to do with that. That's my simplest answer that I can come up with.
That seems like a really good answer.
I mean, I've thought about it a lot. I just haven't put it into the model. And I've thought about a lot of the model that I've got and like how it needs to sort of be started over. Because there's a lot of stuff that I used to do with it very, very well, but there's a lot of new stuff I want to do differently. So I'm sort of grappling at what the right direction is for that.
Thanks.
So
You
it
have some
looks like
questions in the chat.
Hannah has a question about how this would have something to do with clinical studies and injury disease, or how to integrate some of this stuff into the human tissue. And so I think those are really good things to think about in terms of injury and disease. It could be changing perhaps some of the geometry or some of the mechanics or some of the stiffness values, depending on, as muscles are injured, usually something gets laid down with a collagen response to try to add some mechanical integrity to the system while it heals. Sometimes it stays there, sometimes it goes away. So whether that would be changing the mechanical characteristics of just the filaments, or trying to put something back to like what I was thinking about with maybe that parallel passive element that had, like in that hill model, even as an example, if I were to go back to that. I'm trying to share, but I'm doing it really badly. So, even thinking back to that first thing I wrote about the hill model, a lot of what I just showed you for those spatially explicit models were essentially something to do with contraction of a lot of these contractile elements and series elements kind of all lined up together. If you were to imagine that as like what the cross bridge was doing along the thick filaments. But then across the whole sarcomere, there might be something that is a deposit of collagen or scar tissue, for example. Or maybe you could imagine there was an additional element coming off the end of the half sarcomere that represented the tendon, and maybe there was differences in the muscle tendon dynamics or the muscle tendon characteristics with injury versus disease. So there's a lot of different ways that you could try to model that, Hannah. And that's one of the like really cool things about the computational modeling, but one of the more difficult things is there's a lot of ways you could do it, but what's the right way to do it? And sometimes the right way to do it is to just try to get started somehow. And it's no problem to be novice with modeling. I mean, it's good to think about these things and to try to just learn and think about where you might apply some of those parameters to represent the injury, to represent the disease. Like as on the molecular level, for example, if there was something to do with changes in any of the mutations that affected thin filament activation due to troponin and tropomyosin, or changes in mutations in myosin that affected, maybe it was prolonged cross-bridge attachment, so it didn't attach as well. You could try to do a lot of that with changing the kinetic characteristics of what you have in the system if you were trying to simulate some of the molecular mechanisms of disease. So there's a few different scales you could tackle that on. And whether it was in humans or other species, a lot of the components I showed you today are very similar at the molecular and sarcomere and fiber level.
I have another question kind of going off of what you said. So you were mentioning changing some of the mechanics behind the tropomyosin and troponin, I believe. And so do you think that is something that is going on? I hear a lot about gene replacement therapy or gene editing. Is that kind of more or less what you're getting at? And are people using those to integrate into disease-related states, or yeah?
People might be. I haven't seen it very much just yet. And I haven't seen it very much just yet with any of the spatially explicit modeling. There's some more similar, like differential equation modeling, like the actin and myosin cross-bridge interactions, with assuming sort of how those system of chemical reactions, you can write that similarly for the chemomechanical interactions that represent actin and myosin. I've seen it a little bit with that, trying to simulate the effects of different mutations. But I haven't seen a lot of the implications of like a gene therapy showing how that would improve something, either from like the computational or the experimental side, that obviously is illustrating it from a theoretical and mathematical perspective.
Okay, yeah, I was just curious. You know, these are just little things that pop in my mind and snippets from all of the other lectures. So I'm just curious. Thank you.
No problem. I saw somebody else had a hand up there for a little while, but I didn't know who it was.
I might jump in with a broader question while we wait for that person to raise their hand again if they're interested, or put their question in the chat. I was wondering, sort of, Hannah's questions made me think more broadly about, I was wondering if you could share how you think about deciding the right level of, or right kind of modeling approach for a given question, because a lot of the folks here might be new to computational modeling, and within IMSI we have people who are developing computational models at very different levels of organizational scale, from molecular to whole body. So I was wondering if you could share some of your thoughts about how you kind of reason through the choice of model level for a given question.
So I can totally answer that. The one thing that, so to think about the choice of model question, is you sort of need to think about the question you're trying to ask. And you would like to have whatever your model is doing to have some parameter or some subsystem or some portion of what you're trying to describe represented by the process at that level or across those multiple levels. So if you wanted to ask a question about like what's going on as tendon characteristics are changing versus the level of activation in a muscle, well, there's a few different ways you could tackle that, but you'd for sure have to be thinking about some of the mechanical characteristics of the tendon and mechanical characteristics of the muscle, and then some of the activation dynamics. So you need to put a description of the system of what you're trying to tackle. Because if you don't have a parameter that you can test, or if you don't have like a control knob in your model that you can change to try to show some effect of that control knob on your outcome, then you're missing what you need to include. And what's really hard is figuring out how much to include and how little to include, and how to communicate across the different scales, which is why I think there's a lot of excitement to try to close the gap on that with the modeling core group, but I don't think there's been a lot of progress made on that because of some of the differences on the scale and the differences in the complexity there. So one of the tiny things I think we've made some progress on largely has to do with what Kisa's been doing with Rick Kasler. And when she and I have been working together on that, a whole lot of what I've been trying to describe in there or make sure we have in there is some of the components that would go on related to actin and myosin interactions and effects of sarcomere length or filament overlap and passive and active contributions of the sarcomere. And a lot of what Rick and Kisa have been trying to do is sort of wrap that into a higher level. So we've been coming at it sort of from different sides. And I think because I think there has been some really good progress on that concept, but that doesn't have very much to do with any of these spatial mechanisms, because if we wanted to put the spatial mechanisms into that, they're a lot more computationally demanding. And you start to run into problems if you're not doing things in a clever manner, that it takes a lot for your models to run, or a lot of time or computational time. And so the balance between the complexity and the simplicity is where I think the hard part really shows its head up.
Yeah, so if I chime in maybe here, I think like the hope is that that you might be able to derive some, simple rules from the spatial level that could be implemented relatively simply at a higher level without having to, you know, have every simulation go all the way from the nanometer to the, higher level scales every single time. And I'll just say, like, Manny and Miles and I have been playing around with like adding a tendon on, and man, oh man, does that have a really big effect on the viscoelastic behavior. So we're gonna have an interesting conversation about that later today, but it's really, like, yeah, every time you add another level, things get more complicated in unpredictable ways. And but I think what's exciting for me, and I think I speak for Bert as well here, is we've just been thinking about things in a very kind of quasi-static way, and like opening it up to the dynamic aspects just really is exciting. So we have a path forward to make the models more realistic and more applicable to real world movement.
Very cute dog, Monica.
She decided that it was time for me to pay attention to her.
Makes sense.
Go ahead.
I was just going to ask, is there a way that these models can kind of look at like fatigue and different kind of, I don't know, more systems level changes that might affect the lower level mechanics of these molecules, or is that something that's, still down the line? If that question.
I think it might depend on what characteristic of fatigue you were interested in putting in there. My brain jumps to some of the changes that go on with fatigue in terms of increasing phosphate levels, and I think, is it increasing pH levels with fatigue that would maybe have an effect on how the actin and myosin interactions would go through their cycle? But that's sort of, I might be biased in my response that it's a lot of things at the molecular level that I'd think about modeling, but I think there's other things with fatigue that maybe are at the higher level that I don't know as much about. So it would depend on what those characteristics might look like in terms of, I don't know if you'd change your energetics of the system in terms of what fatigue is having an effect on. You might change some effects of what's going on with your excitation contraction coupling, but I'm not very knowledgeable on that. There might be some impacts on the mechanical characteristics of either the tissue or the fibers, though I'm not sure that's known or not known, but maybe it is. So I think it has to do with what property of fatigue you'd want to be adding in. Very similar to some of what maybe I said to Monica's broader question, is what are the properties that we're trying to ask questions about at which level?
That makes sense. Thank you.
So I don't know. I hope that was sort of somewhat informative or a little bit helpful for a different perspective. I know there's a lot that everybody's been covering this summer. Some of it seems easier to grasp than others, so I hope it wasn't too complicated.
This was great. Thank you so much. I learned a lot.
Yeah, for sure we'll be trying out your code.
I am going to post
Did you want me to reload the ending slide?
Oh yeah, that could be helpful too. I'm gonna post the link in the chat too, but if people are in watch parties, the QR code might be helpful. I'll post the feedback form in the chat. And then if people joined late and want to sign in, you can use that other QR code as well.